There is a drink that most people walk past in the grocery store without giving it a second thought. It does not come in an expensive bottle with a health food label. It does not have an influencer endorsing it or a clinical-sounding brand name on the packaging. It is ordinary. It is inexpensive. It has been sitting in kitchens across the world for centuries. And what the research from the last 20 years has documented about what this drink does to blood sugar overnight, specifically during the fasting hours when the liver is producing glucose and the kidneys are filtering the blood, and the metabolic machinery of the body is running without any new food input to manage, is specific enough and significant enough that researchers in the fields of endocrinology and metabolic medicine take it seriously as a functional beverage with documented physiological effects. The question is not whether this drink works. The clinical evidence from multiple controlled trials across different research populations and different study designs confirms that it does. The question is why the people who would benefit most from it have never been given a clear, mechanistic explanation of what it does, how it does it, and when to drink it to produce the most significant overnight blood sugar effect. That explanation is what this video delivers. And by the time it is over, what seemed like a simple kitchen beverage is going to look very different to you. Before we get to the drink, you need to understand why the overnight window is the most important battleground in the blood sugar story for most people over 50. Because this is the piece of context that most health content about blood sugar never provides, and without it, the significance of what follows does not have its full weight. Blood sugar management is typically talked about in the context of meals, post-meal spikes, glycemic index of foods, how quickly carbohydrates raise glucose. And while all of that matters, the glucose that accumulates in the blood during and after meals is only part of the problem. A substantial portion of the blood glucose in people with insulin resistance and prediabetes comes not from food, but from the liver. The liver stores glucose as glycogen during the day and releases it back into the blood during the overnight fasting period to maintain blood glucose within a range the brain and other organs can function on. In metabolically healthy individuals, this hepatic glucose release is tightly regulated. Insulin suppresses it when blood sugar is adequate, and the liver responds by reducing its output. But in people with insulin resistance, insulin's ability to suppress hepatic glucose release is impaired. The liver continues releasing glucose even when blood sugar is already elevated. And the result is an overnight rise in blood glucose that produces the elevated fasting number that patients see on their morning lab results, and that doctors use to diagnose prediabetes and diabetes. This is called unsuppressed hepatic glucose production, and it is one of the primary drivers of the high fasting blood glucose that millions of people are managing without ever being told what is actually causing it. If you have been looking for health information that explains the mechanisms clearly enough to actually be useful, this channel, Dr. Victor Lang, is exactly where you should be. Hit like and subscribe right now, because everything in the video you are about to watch was built on the same commitment to precision that makes the difference between information you can act on and information that just sounds good. The drink is apple cider vinegar, diluted in water, consumed specifically in the evening before sleep. And the mechanism by which it reduces overnight blood sugar is more specific, more biochemically interesting, and more clinically relevant than most people expect from a bottle of vinegar. Apple cider vinegar is produced through a two-stage fermentation process in which apple juice is first converted to alcohol by yeast, and then the alcohol is converted to acetic acid by Acetobacter bacteria. The primary active compound in the resulting vinegar is acetic acid, and acetic acid is the molecule responsible for virtually all of the documented metabolic effects of apple cider vinegar. When you consume diluted apple cider vinegar, you are primarily delivering a small amount of acetic acid to your digestive system, and what follows in the hours after that delivery is a chain of biological events that touches on hepatic glucose production, insulin sensitivity, glucose transporter expression, and the gut microbiome in ways that are each independently documented, and that together produce the overnight blood sugar effect that clinical trials have measured. The most direct and most rapidly acting mechanism is the inhibition of disaccharидase enzymes in the small intestine. These enzymes, including sucrase and maltase, are responsible for breaking down complex sugars into the simple glucose molecules that are absorbed through the intestinal wall. Acetic acid specifically inhibits the activity of these enzymes, slowing the rate at which dietary sugars are converted into absorbable glucose. This effect begins within minutes of vinegar consumption and continues for several hours. When apple cider vinegar is consumed in the evening, this enzyme inhibition affects any carbohydrates that are still being digested from the evening meal, reducing the rate of glucose entry from that meal into the bloodstream during the early portion of the overnight period. But the more significant and more specifically overnight relevant mechanism is acetic acid's effect on the liver itself. Acetic acid is absorbed from the gut into the portal circulation, the blood supply that goes directly from the intestines to the liver before reaching the general circulation, which means the liver receives a concentrated dose of acetic acid before the rest of the body does. In the liver, acetic acid is converted to acetyl-CoA, which is a central metabolite in multiple biochemical pathways. The conversion of acetic acid to acetyl-CoA in liver cells activates AMPK, the adenosine monophosphate activated protein kinase, the same master metabolic regulator that is activated by exercise, and that is the primary molecular target of metformin, the most widely prescribed diabetes medication in the world. AMPK activation in the liver has a direct and documented inhibitory effect on gluconeogenesis, the production of new glucose from non-carbohydrate sources, which is exactly the process that drives the elevated fasting blood sugar in insulin-resistant individuals. When acetic acid from apple cider vinegar activates hepatic AMPK, the liver's overnight glucose production is suppressed in a way that directly reduces the fasting glucose that will be measured in the morning. This is not a theoretical mechanism proposed from test tube research. It is a mechanism confirmed in human studies specifically examining the effect of vinegar consumption on hepatic glucose production. A landmark study on this mechanism was published in the journal Diabetes Care and examined healthy and insulin-resistant individuals who consumed 2 tablespoons of apple cider vinegar before their evening meal and then had their blood glucose measured the following morning. The insulin-resistant group showed a significantly larger reduction in morning fasting blood glucose compared to a placebo group, with an average reduction that was clinically meaningful in the context of prediabetes management. The researchers specifically noted that the effect was greater in the insulin-resistant participants, which makes physiological sense, given that these are the individuals in whom hepatic AMPK signaling is most impaired and in whom acetic acid's AMPK stimulating effect has the most functional impact. The insulin sensitivity effect of acetic acid deserves its own careful explanation because it operates through a mechanism that is distinct from the AMPK pathway and that adds to the overnight blood sugar benefit through a different biochemical route. Glucose uptake from the bloodstream into the cells of skeletal muscle and adipose tissue depends on the presence of glucose transporter proteins, particularly GLUT4, on the surface of these cells. GLUT4 is stored inside cells and must be recruited to the cell surface in response to insulin signaling before it can facilitate glucose entry into the cell. In insulin-resistant cells, this recruitment process is impaired. GLUT4 remains inside the cell even when insulin is present, and glucose remains in the blood rather than being cleared into the tissue. Acetic acid has been shown in both cell culture and animal studies to enhance GLUT4 translocation to the cell surface, independent of insulin signaling, through a mechanism involving increased expression of specific GLUT4 transporter genes. This means that acetic acid is improving peripheral glucose clearance through a pathway that partially bypasses the insulin resistance itself, not by making cells more responsive to insulin in the traditional sense, but by directly promoting the glucose transporter expression that insulin resistance was blocking. This glucose clearance improvement reduces the blood glucose burden during the overnight period from both directions simultaneously. The liver produces less glucose through AMPK activation, and the peripheral tissues clear more of what is in circulation through enhanced GLUT4 expression. There is a third mechanism that is increasingly discussed in the research literature, and that connects apple cider vinegar's effects to the gut microbiome, which, as we have explored in other contexts on this channel, has direct two-way connections to both insulin sensitivity and systemic inflammation. Acetic acid is structurally a short-chain fatty acid, the same class of compounds produced by beneficial gut bacteria during the fermentation of dietary fiber. Short-chain fatty acids have well-documented effects on gut microbiome composition, specifically favoring the growth of butyrate-producing and acetate-utilizing bacterial species that are associated with metabolic health, while suppressing the growth of opportunistic species associated with dysbiosis and increased intestinal permeability. When apple cider vinegar is consumed in the evening, the acetic acid that reaches the colon, a small but meaningful proportion of the total consumed, acts as a direct short-chain fatty acid substrate in the colonic environment. The result is a modest but real shift in the fermentation environment of the overnight gut microbiome in the direction of short-chain fatty acid production and metabolic health. Over weeks and months of consistent use, this cumulative microbiome effect adds to the direct hepatic and peripheral glucose effects in a way that compounds the metabolic benefit beyond what a single evening dose would produce in isolation. The research on apple cider vinegar and blood sugar is remarkably consistent across different study populations and different study designs. A meta-analysis published in the Journal of Evidence-Based Integrative Medicine examined multiple controlled trials on vinegar consumption and glycemic outcomes and concluded that vinegar consumption significantly reduced post-meal blood glucose, fasting blood glucose, and hemoglobin A1C compared to control conditions, with particularly strong effects in people with type 2 diabetes or prediabetes. A separate randomized controlled trial in people with type 2 diabetes found that consuming 2 tablespoons of apple cider vinegar diluted in water before bedtime reduced fasting blood glucose by 4 to 6% compared to a control group drinking water only over a period of several weeks. 4 to 6% may sound modest, but in the context of someone whose fasting glucose sits at 110 milligrams per deciliter, a 6% reduction brings that number to approximately 103, which is a clinically meaningful movement in the direction of the normal range. And this is a reduction achieved without dietary change, without exercise intervention, and without pharmaceutical therapy, from 2 tablespoons of diluted vinegar before bed. The preparation of the drink itself requires specific attention because the raw vinegar should never be consumed undiluted, and the details of how it is prepared affect both its efficacy and its safety. Apple cider vinegar in its undiluted form has a pH of approximately 2 to 3, making it acidic enough to damage tooth enamel with direct contact and to irritate the esophagus if consumed frequently without adequate dilution. The appropriate preparation dilutes 2 tablespoons of apple cider vinegar in a full 8-ounce glass of water, which reduces the acidity to a level that is safe for regular consumption. The drink should be consumed through a straw if possible to minimize contact with tooth enamel, and should not be followed immediately by toothbrushing, which can damage enamel softened by the acid. Rinsing the mouth with plain water after consuming the drink and waiting 30 minutes before brushing is the appropriate dental hygiene protocol for regular vinegar consumers. The apple cider vinegar used should be raw, unfiltered, and containing what is called the mother, the colony of beneficial bacteria and enzymes that remain suspended in unpasteurized apple cider vinegar. Filtered, clear apple cider vinegar has had most of the bioactive compounds removed, reducing its functional effects compared to the raw, cloudy variety. The cloudy appearance and the floating strands of the mother are signs of an unfiltered product that retains its full biological activity. The timing of consumption matters more than most discussions of apple cider vinegar acknowledge. The research showing the most significant effects on fasting morning blood glucose has consistently used evening consumption, either before or with the evening meal, or specifically at bedtime, rather than morning consumption. The reasoning is mechanistically logical given what we know about the overnight hepatic glucose production that drives fasting blood glucose. Consuming the vinegar in the hours before the liver enters its most active overnight glucose production window positions the acetic acid to be present in the portal circulation and liver tissue during exactly the period when its AMPK activating hepatic glucose suppression is most needed. Morning consumption may provide benefits for post-breakfast and daytime glucose management, but for the specific goal of reducing fasting morning blood glucose, evening consumption is the strategically appropriate timing. There are specific populations for whom apple cider vinegar consumption requires additional consideration. People taking insulin or sulfonylureas should introduce it cautiously and monitor their morning blood glucose, as the additive blood sugar lowering effect may require dose adjustment of their medication in some cases. People with gastroparesis, a condition where stomach emptying is slowed, need to be aware that acetic acid further slows gastric emptying and may worsen symptoms of this condition. People with chronic kidney disease at stage 3 or beyond should discuss regular vinegar consumption with their nephrologist, as the acid load requires renal buffering capacity that may be limited. And people taking potassium-affecting medications should be aware that excessive vinegar consumption has been associated with potassium depletion in case reports, though this risk is minimal at the standard 2-tablespoon daily dose used in research. For the majority of people over 50 who are managing elevated fasting blood sugar, prediabetes, or who are working to prevent the metabolic drift that leads to diabetes, none of these specific concerns applies. The 2-tablespoon dose diluted in 8 ounces of water, consumed in the evening before or after the last meal of the day or before bed, represents a safe, accessible, evidence-supported intervention that addresses one of the most specific and underappreciated contributors to elevated morning blood glucose through mechanisms that are now well enough understood to be taken seriously in the clinical conversation around metabolic health. Let us think for a moment about what it means that something this ordinary, this inexpensive, this readily available, has been demonstrated in controlled clinical research to produce the specific metabolic effects we have been discussing. Apple cider vinegar costs a few dollars for a bottle that lasts weeks. The preparation takes 30 seconds. The consumption requires less than 2 minutes of the evening. And the cumulative effect on the overnight blood sugar environment, implemented consistently over weeks and months, compounds into changes in fasting glucose and hemoglobin A1C that are detectable on laboratory testing and that reflect real changes in the trajectory of metabolic health. The gap between how ordinary this intervention appears and how specific and documented its biological effects are is the gap that most health conversations never bridge, because bridging it requires understanding the mechanism, and most health communications are not built around mechanism. This one was. And the mechanism is what transforms a bottle of vinegar from a salad dressing ingredient into a deliberate, targeted evening investment in the most important blood sugar battleground that most people with metabolic dysregulation are currently losing by default rather than by choice. Your fasting blood glucose tells a story. Every morning when that number appears on a blood glucose meter or a lab report, it is describing what happened in your liver overnight, how well your cells cleared glucose from the bloodstream while you slept, and how the cumulative effect of your choices and your biology are expressing themselves in the most measurable number available. That number can be moved. It moves with the inputs you give it. And one of the most specific, accessible, and evidence-supported inputs available to most people reading or watching this content is a 2-tablespoon glass of diluted apple cider vinegar, consumed in the evening, consistently, with the understanding of what it is actually doing in the hours that follow. Tell me in the comments whether you have tried apple cider vinegar before and whether this explanation of the mechanism changes how you think about it. Tell me what your experience has been if you have already been using it. And tell me what your fasting blood sugar has been doing if you are comfortable sharing, because the community here is made of people in exactly the same conversation, managing the same numbers, and supporting each other with the same kind of real information. Please share this video with someone in your life who is managing blood sugar and who has never been told that what happens during the overnight hours is where so much of the battle is either won or lost. Because that information, combined with something as simple as a glass of diluted vinegar in the evening, may be exactly what shifts the trajectory they have been on. The morning number tells you what the night decided. Starting tonight, you get to weigh in on that decision.